Okay, so today we're going to go more into the ecological or environmental aspects of microbiology. So we're going to look at microbial ecosystems. Now, before we get into some of the details of microbial ecosystems, first thing we have to do is kind of introduce you to some of the general terminology. Now, what I did with this slide is I gave you a nice big figure over here. This is because so much of what we focused in micro up until now, or what we generally focus in micro, is a lot of cellular science, the molecular, cellular side of things. But I kind of want you to see the big picture now and kind of let you see how ecology relates to the cellular aspect a little bit better. That way you'll be able to understand the significance of microbial ecology and ecosystems. And what I mean by the big picture is look at how you can see the ecological aspects up here, like the biosphere and the ecosystems, and then how it connects down here to the more cellular and molecular side of things, with molecules and cells being those smallest subunits that we're going to see on that figure. Now, when we get into the idea of microbial ecology and ecosystems, there's two terms I want you to know. There's ecosystem and there's habitat, and both are very significant. Now, the difference between them is when you hear the word ecosystem, that's the sum of all of the organisms and the abiotic factors of a particular environment. So that means it's living and non-living things in an environment, because that's what we mean. All organisms are living, and abiotic factors are non-living things like the air, the water in that environment. Now, when you have the term habitat, what that means is a portion of that ecosystem that we just talked about where the community could reside. What that's a fancy way of saying is the place where those organisms will reside. Okay, so habitat is part of ecosystem. That's how they fit into the grand scheme of things, is the idea that an ecosystem will contain many habitats. And when you think of those ecosystems, it's made up of, the community and the non-living things. And when we say community, those are populations of organisms, right? Multiple populations living together. And then when you look at the figure, when we say population, we mean multiple of the same type of organism. And then when you zoom in on just one of them, now that's the organism. And when you break down that organism, now we get into the cellular type of things, like tissues and organs and the systems that you go through in A&P class. And then you break those down into the individual cells and finally the little old molecules. Okay? Now, when you ask yourself, where do microbes fit in this big picture of things, from down here at molecules all the way up to biospheres? Well, the answer is microbes fit in at every single level except for molecules. Okay, so you find microbes all the way from cells up to the biosphere. Okay, so microbes are a very big part of ecology. Now, this is just a recap slide, so you can write down the definitions of the terminology in case you missed them. Now we get into a little bit more terminology, and all of these are terms that you've seen before, not just in our course, but most likely in many other biology courses. This is the idea that, microbes, they establish a lot of different relationships. A lot of relationships with each other, but also with other organisms. And when you have those relationships amongst organisms, we call that symbioses. You also hear it as symbiosis, okay? Now, these relationships between organisms, they can have various different categories. You can have parasitism, which I always refer to as the, professor in undergrads. Parasitism is where one benefits and the other one is hurt. So whenever you hear in microbiology a pathogen, for instance, pathogens are a parasitic symbiosis with the host. They're hurting the host for their own benefit. If, however, both organisms are benefiting, such as your normal flora and you, then that's called mutualism. So for instance, those bacteria in your GI tract, like E. coli, that help you digest foods, especially, the cellulose of vegetables, that is mutualism. Then you have commensalism. And commensalism is a little tougher to have examples in the human body sense. Usually you see it more out in ecology, especially with fungi. Commensalism is where one of the organism benefits, but the other one's completely unaffected. Now, on the figure below, they use staph epidermidis, but I don't like that example for commensalism, because when you think of staph epidermidis on your skin, well, technically that's mutualism, because the staph is getting the benefit of feeding off of, your lovely skin and all of that, beautiful stuff there. But, the staph epidermidis, it's getting a nice home. Whereas at the same time, it's helping us in the sense of taking up space and resources so that pathogens won't get us. Because remember, staph epidermidis is part of your normal flora. So I don't like that example. Instead, when you think of commensalism, think of a lot of fungi. Fungus outdoors loves, the commensalism type of relationship. Okay, so make sure you understand the difference between parasitism and mutualism, and then commensalism as well, and always kind of be able to give yourself an example of each. Now, when we talk about microbial ecosystems and microbial ecology, you have this concept of the diversity of microbial species. Now, there's two terms on this slide that you have to know. There's species richness, which simply means the total number of different species in that ecosystem, versus species abundance, which is the proportion of each of those species. Okay, so when you think of it, species richness is the total number of all the different species, whereas species abundance is now looking at each individual species and seeing how abundant it is in that ecosystem. So the proportion of that species versus, let's say, other species there. Okay. Now, when you talk about this, it both kind of depend on what's available to them in that environment. So kind of like when we talk about your normal flora and their environment, they're using up resources and taking up space, and so that limits other potential bacteria. Well, the same thing happens in microbial ecosystems, where if certain species are taking up or better at taking up, space and resources, then that may limit the other species that can be in that ecosystem. Now, within that ecosystem, you'll also hear the term species diversity. That's simply the number of species and percentage of each in comparison to the total. So that's the overall species diversity of that ecosystem. Now, when you think of the microbial ecosystems, you also have to think of what we call biogeochemistry and nutrient cycles. So I'm sure you've heard the term nutrient cycles before, but you may not have heard biogeochemistry. Okay, what we mean by that crazy term biogeochemistry, yeah, I love to say that, those are the biogeochemical cycles that transform key elements in that ecosystem by biological and chemical agents. So basically, that what I'm saying there is that these cycles are responsible for things like redox reactions, respiration, fermentation, even the nutrient cycles themselves. Okay, so these are all terminology you've heard before, but the overall umbrella term for those reactions that are occurring is biogeochemical cycles. Now, within this ecosystem, you also have the terms guilds and niche or niche. The guilds, what that means is all of the metabolically related microbial populations that are interacting with microbes and the abiotic factors of that ecosystem. Okay, so one more time. The metabolically related microbial populations are interacting with other microbes as well as abiotic factors, so living and non-living things. Now, the niche, what that is the habitat shared by those guilds. Okay, so the niche is the supplies, the nutrients, even the conditions required for growth, okay, that these species may need. Now, what you see in the figure here is giving you an idea of the various guilds that you'll see within one single community. And you have all of these microbial communities that will be near each other, interacting with each other, but also interacting with cycling energy and things like nutrients. Okay, and notice that a single community will have multiple guilds within it, multiple of these microbial populations interacting with abiotic factors to kind of help that ecosystem, to help break down nutrients and to cycle through things like energy. Here's another terminology recap so you can pause and take down the exact definitions that I was just talking about. Now, when we talk about microbial ecosystems, you have to think about what the microbial environment is. And there are two terms that I want you to know for this highlighted here. They are microenvironments as well as the different types of niche. So you have here first microenvironment. When you break down that word, micro is really small and environments is the area around an organism, right? So a microenvironment, what that's referring to is when you think of microbes, they're tiny. They are so small that when they directly, experience life around them, if you look at it from the big picture, that tiny little microbe is only going to experience a really small amount of the local environment, right? Because that microbe, for instance, if it's messing around in your body or on your laptop, well, it's never going to see, it's never going to make it a far, far distance away and experience, let's say, all across the nation, the way a person might be able to. Usually, because they're so small, they're only directly experience a very small piece of their actual environment. Okay, so the microenvironment is the tiny local environment that a microbe ends up experiencing. Now, the next terms that you have here are associated with niche. So realized or prime niche, what that's talking about is when you have a niche, you have, resources and conditions that an organism will require or need, and they'll need it in different types of quantity and whatnot. There's always going to be at least one niche in which that organism is most successful, especially because there's so many limiting factors working on that organism. So that one main, actual niche or conditions and resources that microbe will actually live in, That's called the realized or the prime niche, okay? The actual resources and conditions for that microbe. That's as opposed to what we call the fundamental niche, which is all of the conditions or resources in which that microbe would be able to survive, okay? So it's kind of like when you think of optimal versus, what optimal temperatures and whatnot versus where a microbe actually ends up, surviving and living out its little life, okay? I also want to point out that when we talk about these conditions and resources for a microbe to survive, you have to keep in mind that these conditions can really quickly change, okay? Think of, even what you've experienced in your life when a pandemic suddenly pops up, okay? Microenvironments can rapidly change, and so you end up with what we call feast or famine existence for microbes when they're out in nature. It's basically saying that resources will always enter that environment, but they'll quickly get depleted. Think of, how much bacteria eat, how much they cycle through resources, how quickly they replicate, right? So feast or famine in nature means that as the resources enter that environment and those microbes get access to resources, those resources quickly get depleted. So you get them cycling through these times where they have a lot of resources available and then they use them up. So they're in like a famine time, but then more resources enter. So for instance, a bird drops dead out of the sky, lands right there on the ground. Well, now those microbes that are there in that area have a beautiful feast, but they're going to break down that dead bird, as is, the rest of the environment around it is going to deteriorate that dead bird. So that's only there for them for a limited amount of time. And so then it'll get depleted, but something else will occur. Now a little mouse dies over there. So now there's more feast, but then they deplete it. So you get this cycling. The other thing I want to point out is that growth rates, because of that, especially that feast or famine, end up lower in nature versus the lab, because they're typically going to grow in spurts based on that feast or famine. Are resources available? Are they depleted type of situation that they're in? Because in nature, conditions are rarely optimal. So their growth will depend on the availability and the nature of the resources that they encounter. Whereas a lab, you're giving them all the food they want. And a lot of times you even dilute them down, give them more media, take them away from their wastes and whatnot. So a lab, you see higher growth rates. Okay, so put a little star next to that. Make sure you understand, what is meant by feast or famine, as well as why, microbes end up with growth rates lower in nature versus a lab. So I could ask you, in which condition would the microbes have a lower growth rate and why? So here's another terminology recap for you to pause and jot down some notes. I did not write the notes on feast or famine and growth rates because that's not terminology, that's more conceptual. So make sure you also emphasize those as well. Okay, here we have our old friend biofilms. So biofilms are not just something that you find in your mouth on your teeth. As we mentioned before, they're actually quite common even in the medical field and in the microbial environment. Because when you think of the microbial environment or any kind of environment, there are a lot of surfaces that are perfect for a bunch of microbes to stick together, gather together, and slime up basically. So when you think of surfaces, you have to realize that they're a great location for biofilms to form because there's plenty of nutrients. It allows for the bacteria to then gain protection, and it's a nice location for them to stick. So for instance, when you think of rocks in a pond or lake or any water area, that's a great surface for microbes to build their own little community there. So now when you think of biofilms, they're basically going to gather and collect on this surface, because we've mentioned that biofilms are a collection of different species of bacteria that clump together, stick together, and they form this big population together. Now, on the figure on this page, you can see how exactly biofilms form. So when biofilms form, there are 4 stages. The initial microbial cell will attach to the surface, and then you get colonization, where you get these multiple microbes now producing polysaccharides. And as you know, polysaccharides, that's like sticky sugars. Basically, a lot of glycocalyxes, remember that slime layer or capsule, that can be around certain bacteria. You then have the development stage of the biofilm, where you're getting more and more and more bacteria clumping up, and you're getting more polysaccharide formation. Lastly, you have active dispersal, and that's basically triggered by the environmental factors, and that's where some of those bacteria will now break free and disperse throughout that environment and can now go start another biofilm nearby. Now, when we talk about biofilms, you have to think about, why would a bacteria want to form a biofilm? Why would they want to be part of this big community of other different species? Well, there are a few reasons I want you to think of, okay? 3 main ones. The first one is self-defense. If you're in part of a biofilm, whether it's on teeth or whether it's out on surfaces in a rock, in a lake, wherever, you now resist physical forces better, especially any physical forces that might sweep away unattached cells, okay? So even things like toxins or antibiotics might be less effective on you if you're part of this big clump of a biofilm. Secondly, a biofilm allows the cells to remain in a favorable niche, meaning that it allows these cells to be part of a group now that has good nutrients and conditions for growth. Lastly, it's good in terms of living in a close association, basically having friends or backup in terms of getting things done better. So think of something like quorum sensing, how if you're part of a biofilm, it's more beneficial for you. Now in this slide, I just want to give you some examples of biofilms. We've already mentioned multiple times the medical and dental examples. So the idea that plaque on your teeth is an example of biofilms. And with that, you end up with a lot of dental conditions such as periodontitis, gingivitis issues. You also end up with a lot of medical complications from biofilms. You can get them in a lot of hospitals or medical facilities, especially they like surfaces like catheters or IVs. So you end up with various infections that way from these biofilms. Plus, a biofilm like plaque on your teeth can spread to other locations. So for instance, the bacteria that collect on your teeth happen to be one of the biggest causes for heart conditions, especially endocarditis. Okay, so these bacteria can travel to your heart, they can travel even to your brain and have been linked to dementia issues and Alzheimer-like symptoms. Okay, so you have to take biofilms very seriously, even though people, think, oh, everyone has plaque, it's fine. Do be careful. Be careful about, scratching at your gums. Be careful about how hard you floss, or even when you go to the dentist, you have to take these things into consideration. You also have biofilms, like we just mentioned, the idea of forming on surfaces in the environment. So you get a lot of biofilms in the industrial sentence, in places like in pipes. And these can end up causing problems like corrosion on these kinds of surfaces. And the last one that I mentioned here is microbial mats, which are basically really thick biofilms. You find them especially at the bottom of shallow ponds, where you get a ton of these mats, where you get a whole collection of different bacteria that are usually phototrophic or have abilities like chemolithotrophic abilities. So if you remember phototrophic, they're able to use sunlight. Chemolithotrophic, these are organisms able to use inorganic matter such as CO2. And so you have these collection of bacteria that can produce energy and carbon from things like light sources and CO2. So you end up, they have this perfect environment for those kinds of abilities, and you get very thick, dense collections of them. Okay, so make sure you know some of the examples of biofilms other than plaques. Now, when we talk about microbial ecosystems, I also want to mention terrestrial environments. So everybody's heard the term soil, but you may not have really thought about what soil is. You can have mineral soils or organic soils. So with mineral soils, these are soils from rock weathering and very heavily inorganic materials. Whereas organic soils, as the name suggests, it's organic, derived from sedimentation in bogs and marshes. Now, most soil is actually a mixture of both mineral and organic soils. So again, mineral being inorganic versus organic soils, of course, being organic. So a lot of soils are mixtures of both, and you end up with this environment of inorganic material at around 40%, organic material being around 5%, and then you have a whole bunch of air and water being around 50%, and living organisms being around 5% in this terrestrial environment. Okay, but what I want you to remember is the idea that soil you see as mineral or organic. Now, there is a ton of microbial life in soil, whether that soil is mineral or organic. It's a type of structure that really is conducive for organisms to live in. Okay, so of course the biggest amount of microbial activity is going to be where there's most rich surface of nutrients for them, and that happens in the rhizosphere. And where you find the rhizosphere, what that means is the soil that surrounds plant roots is going to be the most robust for organic life in that soil, because around the plant roots, these organisms receive plant secretions, which are great nutrient sources for these microbes. Now, ultimately, the big factor affecting whether microbes can inhabit certain areas of the soil is going to be the availability of water. Now, I know you might picture soil and be like, oh, dirt, that's kind of dry. Where are they getting water in that kind of environment? Well, a lot of soil is capable of holding on to droplets of water that you might not even see with your naked eye. And I like this little diagram here, this picture here, because it helps show that you get in these microbial soils and particles, you end up with the droplets of water kind of stuck between the components of sand and clay and little bacteria all over the place. So this is actually looking at like a soil clump where you would have the water trapped within there, because the microbes are going to need access to some water. So now phylogenetics is a term that you hear a lot in terms of ecology and evolution, but phylogenetic sampling can also involve soil because of all of those microbes that are capable of living within that soil. So phylogenetics or phylogenies, when you hear about that, that's basically when you think of evolutionary trees, all of those who's related to whom in terms of different species. So within phylogenetics, looking at how different species are related to each other or differ from each other, each species can end up being a phylotype if it's defined by a particular sequence that that ends up differing from all other sequences by 3%. But you don't have to memorize those numbers or that terminology. I just want you to be aware that there's a lot of microbial diversity in the soil in that kind of environment. So for instance, here you see all of these different groups of types of bacteria that you might find in a particular soil location. And so when you hear phylogenetics, just think of the diversity of microbes in soil. Now, when we think of soil and microbial life in it, we said that the greatest amount of microbial activity is going to be up in the rhizosphere, near where you have plant and heavy nutrients, for instance. But there's also the deeper surface to the soil, what we call the subsurface of soil. And that can extend way, way down deep into that soil surface, below that soil surface. And because this environment, if you kind of picture it, it's not going to have as much nutrients as the up top portion, and it's going to have a little harsher conditions because think, deep down within the crevices of Earth, that's kind of a scary place. So you end up with a lot of archaea, if you remember archaea being the type of prokaryote that can basically survive in extreme conditions. So for instance, in the figures that you're seeing down there, or in the idea of deep, deep subsurface, archaea are very capable of surviving in areas that may not have the most robust nutrients or water, or may have odd temperatures or pressure. So even for instance, in the figure here, so this one here, when they're sampling hot fissure water in gold mines, they end up finding a lot of archaea, because if you think hot fissure, well, that's, archaea are the only type of cells that would really like that kind of environment. But what is interesting is even though you may picture deep subsurface as having like no nutrients or survivability, there actually do end up being there does end up being access to nutrients deep down there, because of the idea of groundwater flowing through the crevices within the soil. So groundwater ends up giving them some access. And also the idea that hydrogen is thought to be continually produced even in those odd locations because of the interaction of that water with various things like iron minerals, or what we call radiolysis of water. But you don't have to know all about that. Just be aware that archaea especially like the subsurface, and that not all microbes are going to be in the rich rhizosphere, even though that's where most of them will be. So now moving on from the soil, you also end up with a lot of aquatic environments. And the first one we're going to mention is the freshwater location. Now, when you think about water, you have to realize that oxygen has limited solubility in water. So depending on where you're looking at the water, deep layers within that fresh water can end up becoming what we call anoxic. Okay, so you have these conditions of anoxic environments, and so the oxygen concentration ultimately in those aquatic systems ends up dependent on the amount of organic matter present. and the kind of mixing that you get within these water environments. Now, when you think of these kind of environments, photosynthesis is going to be a big deal in water environments. So we've even mentioned the original great oxygenation event happening in water environments way back when, and we wouldn't have human life. We wouldn't have us if it wasn't for the organisms, the microbes that you find in the water environments. Now, one of the important organisms to know in the freshwater aquatic environments are phytoplankton. And what phytoplankton are, these are going to include things like algae, or again, those cyanobacteria that we mentioned for the great oxygenation event. And these phytoplankton, they're oxygenic phototrophs. Okay, so they're able to use light. And they're suspended freely in the water, and they're using this light-driven splitting of water to be able to produce oxygen, which ended up being very valuable to us humans. You then also have benthic species, as opposed to what we call pelagic. Okay, so I have those words written down here, benthic versus pelagic species in freshwater. What that means is benthic species are going to be the ones that are attached to the bottom or the sides of lakes or streams. Okay, so you'll find them basically in the mud. So a lot of little, if you've ever dug up any kind of sand or mud near even the ocean water, anything in the mud or the bottom layers, they're benthic. Whereas anything floating around and swimming around up in the actual water itself, that's pelagic. Okay, so benthic is at the bottom. You can think B, bottom. Benthic bottom in the mud versus pelagic swimming around. Now, with aquatic environments, you also end up with rivers as well. And with rivers, unlike, the fresh water, the more stagnant water that we might have been thinking of a minute ago, the difference with rivers is they have a really rapid water flow. So you get a lot of mixing of that water, which is a great thing for microbes because you don't end up with as much of those anoxic conditions that I was talking about, where oxygen gets used up but doesn't get restored into that, big freshwater environment. Whereas rivers, they're rapidly moving so much that even though organisms will use up the oxygen and you would expect anoxic conditions, instead that water gets moved around and cycled, and oxygen from the top layers ends up coming back down into the lower levels. So you don't get as much of a problem with oxygen. But even in rivers, you can still end up experiencing oxygen deficiencies if there are high inputs of things like organic matter or pollution that ends up in these rivers, which can take its toll in terms of the sustainability of that waterway. Now, within the rivers and the aquatic environments, you should also know what we call biochemical oxygen demand, which we shorten as BOD. Okay, and the biochemical oxygen demand, the way I have it written on this slide, is the microbial oxygen consuming capacity of a body of water. Now, basically, if you think about it, bacterial numbers increase, and as those increase, oxygen levels will decrease. Okay, oxygen levels decrease, and there's a spike in inorganic material. Because if you picture bacteria, all those large numbers using up the oxygen. Okay. Now, when you end up with this situation where the higher bacterial numbers used up or depleted the oxygen levels and spiked the inorganic matter, you then end up with a rise in algae and cyanobacteria in response to the inorganic nutrients, because algae and cyanobacteria, they thrive off of inorganic matter, which might be looked at as wastes. to many other organisms. Okay, so you end up with this important cycling within the river. You then also have oceans as an aquatic environment. And now when you think of the ocean, you kind of have to think of what a vast open ocean really is. And first of all, when you compare it with the freshwater environment, well now, what do you notice? It's very salty, right? High salinity, okay? How is that going to affect the microbes that live in there? Well, they're going to have to be halophiles, okay? So oceans will have a lot of halophilic bacteria. Now, because it's such a vast waterway, and it doesn't have that rapid water flow that rivers have, it ends up being low in nutrients because any of the organisms, they'll use up the nutrients, but it's not moving quickly, and it's such a big area that you don't get quick replacement of those nutrients. It's also because of how vast it is, it's going to end up a lot cooler than the other types of waterways. Because think about it, what warms up these waterways? Well, sunlight. The oceans, sure, the top of them are getting hit with that sunlight, but oceans are deep. They are huge, massive. And so once you get down below a certain level, the sunlight, the warmth, the heat from up top is not going to reach down there. Okay, so the size of the oceans ends up causing a lot of the major factors that are affecting, which microbial activities can actually take place. And if you picture the idea of these vast oceans, I want you to remember that near the shore, the closer you are to the shore, the higher the microbial numbers will be because of how high the nutrient levels will be over there. Whereas the further out you get, there's going to be less, life, less nutrients kind of introduced there. The further down you go, there's going to be colder, less oxygen. Okay, so you end up with a differing variety of organisms depending on where in the ocean you're looking at. And the activity of these organisms will be limited by the factors that we kind of just mentioned, the nutrients and the temperature and all of that. Now, when we talk about oceans and that vast, vast amount of water and surface, you end up with what we call oxygen minimum zones, OMZs. And those are basically areas where the oxygen's been depleted. Because again, think of, where you have organisms living, where you have things using up oxygen, where you don't get any replacement of that oxygen, or where you don't get as high of a replacement for oxygen. Okay, so oxygen, the demand is always going to be high, and sometimes, certain areas can't fulfill that. Now, you end up with what we call high levels of denitrification and anammox. And what that means is anaerobic ammonium oxidation, which is a fancy way of saying you end up with the nitrogen cycle there. Now, these OMZs are expanding, and you have to ask yourself, well, what does that mean for microbial composition, for the types or variety or robustness of microbes that are going to be in this area? Well, think about it. If we're saying that oxygen is getting depleted, well, what does that mean? Aerobes, they can't survive in areas where the oxygen's been depleted, right? So instead, you end up favoring, these OMZs end up favoring anaerobic microbes at the expense of the aerobic ones. Because when there's a depletion of oxygen, anaerobes thrive, aerobes, they don't do well. Okay? Now, what's interesting is that some bacteria ended up evolving to use nitrate instead of oxygen. That's what we meant above by denitrification. Okay, so if bacteria evolved to use nitrate instead of oxygen, and also the anaerobic ammonium oxidation, which means using, the nitrogen cycle, well, then these bacteria will also be favored in OMZs because they don't need oxygen. Instead, they could use nitrate or nitrogen. The last thing to mention here is asking yourself, what does this mean for atmospheric chemistry and for the food webs? So if you think about it, okay, for atmospheric chemistry, atmospheric oxygen and nitrogen are dependent on microbes that basically die in those hypoxic conditions. And so you end up with things like crabs moving higher up on the water column, okay? And oxygen levels end up, depleted. That's going to be intolerable for fish survival. So, with the food webs, you kind of see basically a suffering there from these OMZs, okay? But what I really want to emphasize here is what is meant by OMZ, so that's oxygen minimum zone, and the idea that it ends up favoring anaerobic microbes as well as the microbes that are capable of using nitrate or the nitrogen cycle. So it favors denitrification and that anammox word that I mentioned before. So what I want you to remember when you think about oceans and ocean life is the fact that most of the primary productivity in those open oceans is due to photosynthesis, specifically by prochlorophytes. Okay, so prochlorophytes are basically types of phototrophs. So all of the organisms on this slide, they're using light energy in order to make their carbon sources, or in order to obtain their carbon sources and build all of their metabolic needs. You don't have to memorize these three guys. I just want you to be aware of how significant phototrophs are in the oceans. So notice how, for instance, the first one, Prochlorococcus phototrophs, how they're around 50% of the net primary production in these marine environments. So they're very significant organisms. Okay. Now, still taking a look at the major marine phototrophs, here's another example. And this is now looking at aerobic anoxygenic phototrophs. Okay, so they're using light energy, but they're not fixing carbon dioxide. And so their light usage is more for energy in terms of photophosphorylation. And when we say photophosphorylation, so you can circle that word, photophosphorylation means a light-dependent process, specifically the idea that there's a proton gradient generating ATP from the phosphorylation of ADP and energy of the sunlight. Okay, so photophosphorylation is how we're obtaining energy here. Now, on one of the previous slides, I started to use the terminology pelagic and benthic. So make sure you know those terms. Make sure that on this slide, when you see pelagic bacteria, that tells you that these are guys that are floating around in the water itself, rather than being down at the bottom. So these will be surface water organisms. Now, within these pelagic categories, you have planktonic heterotrophs, and with the when you're looking at the water gradient in these zones, you have to realize that the prokaryotic densities in the open ocean will decrease with depth, which makes sense. Okay, it makes sense that you'll have bacteria mainly up at the surface where nutrients, light, resources are most abundant. Then as you get deeper, think about it, deeper and deeper in that ocean, you're going to have less of these regular prokaryotes, these regular bacteria, because there's less light, there's less oxygen, there's less resources. So bacterial species will tend to be more at the surface, but then when you get lower down, well, now think of it, now you're heading toward environments that are more extreme, more abnormal. So now you're going to have an increase in the archaea. Okay, and that's why we say that as you get deeper, you end up with bacteria and archaea kind of equaling out, and then archaea becoming more as you go even below that level, because up at the surface you have a lot of bacteria and not so much archaea, whereas as you go down, fewer resources and whatnot, less bacteria, but archaea thrive in those extreme conditions. Okay, so make sure you kind of understand the idea of archaea versus bacteria. And interestingly enough, you're going to see in a few slides, there are a lot of viruses in the ocean too. But we're not going to talk about them on this slide. We'll get to that in a minute. Now, what I want you to remember is highlighted in red here, the most abundant marine heterotroph is Pelagibacter. Okay, so whenever you see Pelagibacter, think of that as the most abundant marine heterotroph. It is specifically what we call an oligotroph. So I wrote the definition for you here, since you may have, of course, heard of autotrophs and heterotrophs, but we haven't really used the term oligotroph yet. What that is an organism that grows best at very low nutrient concentrations. And like we said, in the marine environment, you do end up with a lot of areas that are low in nutrient concentration, especially where there's less light, less heat in these areas. So oligotrophs thrive in that kind of environment where, other organisms may not. I also point out here that a lot of these guys are using proteorhodopsin, and that's basically how they're able to use light energy in order to make energy for these cells, in order to make ATP. But we're not going to go through the exact mechanism, just be familiar with them using proteorhodopsin in order to make their ATP. And keep emphasizing to yourself the fact that the bulk of these marine organisms are using light energy. So now here's an interesting fun fact. Viruses are the most abundant microbes in the oceans. And this might be kind of surprising to you, because when you think of viruses, I'm sure you think more of human infections. You're thinking of viruses, swimming around in humans. But the funny part is that viruses are hugely common out in oceans. And these viruses, they're very robust and varied in terms of the types of viruses you encounter, and their presence can greatly affect, the other microbes that you will find out there in the oceans. Now, to give you a little bit of phylogenies, okay, so we mentioned earlier that when you hear phylogenetics or phylogenies, you're looking at the different species of organisms, how they're related to each other, and their abundance in terms of a particular location. So you notice, like we said before, proteobacteria are going to be the biggest group in terms of these phylogenetic breakdowns in the marine environment. Okay, and then the rest is just to give you an idea of various other organisms that you're going to find within that marine environment. But the rest of it, you do not have to memorize all the other names. I just want you to circle proteobacteria. Now, what's interesting is you would think that, all life is just up there at the surface where the light is and where all the beautiful nutrients and all are, where you see all those lovely little fish swimming around. But interestingly enough, the deep sea and the deep sea sediments also have a lot of organisms still capable of living in there. Now, the bulk of the ocean actually is deep sea. It's areas that you can't even see because it's so far down there, and you're just seeing the small surface of the ocean. And I know it sounds funny to say the small surface because to your eyes the ocean looks massive, but the funny part is you're just seeing the tip of the iceberg there. Okay, now when we talk about deep sea, you have to start considering what these microbes or these organisms have to deal with if they're going to live in the deep sea. So think about it. The deeper down you get in these oceans, visualize it, they're not getting much light, right? They now have the absence of light energy because they're so deep down. Now, without that light, they're also going to be a much lower, much more frigid temperature. And that'll ultimately equate to a lot less nutrients. Okay, they're not going to have all that introduction of nutrients that you might get up top, okay, from having, more robust animal life and light up top. So they're going to be no light or very little light. There's going to be very cold temperatures. There's going to be barely any nutrients. And the deeper you go, the higher the pressure is. Okay, so think about when they're doing scuba diving training, how careful they have to be about the pressure change the deeper you go. So I want you to keep in mind all of these conditions that deep sea organisms are going to have to live with. So now with those crazy deep sea conditions, you end up with some very interesting microbes and some terminology that's slightly different from what we've gone over so far. So I want you to know all four of these terms and how they differ from one another, okay, and the fact that these are the main organisms that you will find in deep sea environments as opposed to elsewhere. So the first term you've actually heard me say before, that's psychrophilic. So the psychrophiles, those are the ones that I called psychophiles, the microbes that like really cold temperatures. So psychrophilic microbes are the ones where the cold temperatures are their optimal growth temperatures. Psychrotolerant then is a little different. So what psychrotolerant means is now these guys are more like mesophiles in that their optimal temperature is that medium type of temperature, like room temperature and whatnot, but they are still able to tolerate lower temperatures at slower rates. Okay, so at temperatures that a mesophile would have died at, the psychrotolerant ones, they can still survive. You then have piezophilic and piezotolerant. Now, instead of talking about temperature, we're talking about optimal pressure environment, okay? So piezophilic, these are deep sea microbes that have their optimal growth under high pressure conditions, okay? So these are the ones that would be great during finals week. They do best in high pressure. Then you have piezotolerant. So their optimal growth condition is regular pressure, but they're able to tolerate the high pressure conditions of the deep sea location. Okay, so keeping in mind the difference between philic and tolerant, philic means that that condition is their optimal, whereas tolerant means that condition's not their optimal, but they'll still survive it. Okay, they might do so at a slower, less content rate, but they will still survive it. Okay, so make sure you're very comfortable knowing all of these terms and how philic versus tolerant differ. Now this slide is just to give you a little more of the phylogenetic breakdown on any of these, you don't have to memorize any of these organisms or whatnot. It's just to help you visualize what's going on at the deep sea location. Now, I do want to point out that you notice that proteobacteria is still the most dominant out of all of the different groups on these pie charts. So in deep sea or in the surface area, those proteobacteria are going to be the main marine microbes. Okay, so again, you don't have to memorize this. It's just for you to visualize all of the little organisms that are living deep down in the deep sea. Now, one other thing I want to point out is that you'll notice on this slide for deep sea, now it mentions sediment microbes. So it's saying sediment. Remember that whenever you hear sediment, that means you're talking about benthic organisms, as opposed to the pelagic ones that are swimming around above in the water itself. The last couple of things I want to mention in this lecture involve hydrothermal vents. Okay, so hydrothermal vents, they're going to have warmth or be very, very hot locations. So the organisms that live there are going to have to be okay with those very hot temperatures. So basically think of it as you're either going to have thermophilic bacteria or you're going to have archaea, because archaea love extreme kind of conditions that you or I would just die instantly from. Okay, so with these deep sea hydrothermal vents, you end up with thermophiles or with archaea, and the dominant type of bacteria with regard to how they're getting their food or carbon sources or energy. Now you're dealing with chemolithotrophic bacteria. Okay, so chemolithotrophic means that they use inorganic material to make energy. Okay, and a lot of them will be autotrophic with regard to how they're obtaining carbon. Okay, so chemolithotrophic and autotrophic are going to be the big categories with regard to how they're getting energy and they're getting carbon sources, because if you think about it, when you picture hydrothermal vents, there's not going to be that much in terms of organic material or nutrients because it is an extreme kind of environment. With regard to temperature, we categorize the bacteria that will be living there as thermophiles or hyperthermophiles. So keeping in mind again that hyperthermophiles is a fancy way of saying extreme thermophiles. And the ones that are living there in terms of regular thermophilic categories can either be facultative or obligate. Okay, it doesn't have to be one only. Now, the last thing I want to show you with regard to these hydrothermal vents is the same thing we've been seeing with each of the other locations, and that is their phylogenetic pie charts. Okay, so here's the last of our phylogenetic pie charts. Okay, so you get to see the various breakdown, and again, what are you noticing? Look at that big blue chunk, that's proteobacteria. Okay, so keep in mind that throughout all of the different aquatic type locations that we talked about and the hydrothermal vents, proteobacteria are the most robust that we kind of mention here. But again, the rest of it, you don't have to memorize these pie charts or anything. It's just to give you a visual breakdown so that you can see that even these locations where you wouldn't expect anything to survive, microbes are quite robust. Okay, they can handle a lot of things, whether it's high heat, whether it's low heat, whether it's high pressure, low pressure, all of that, you end up having some organisms survive. Okay. So now it's time for you to head over to the discussion board, and I apologize if some of the material in today's lecture isn't as in detail as some of the other lectures. I will admit the environmental science stuff is not my main area of research and study. So if you need any further clarifications, just send me a remind message as always. Thank you and have a great day.